Thank you. This picture, by the way, was taken about 2 miles from my house. The problem I was given early on about a deer population turned out to be quite close to home and not at the ends of the earth as often has been the case with. And I think I hit something wrong here. Okay, so what I'll talk about today is just a brief history of SpayVac and a summary of SpayVac deer trials, a little bit about how SpayVac works, although that's likely to be redundant given a lot of the talks that have been given here already. Comparison with conventional contraceptive vaccines, and then just a brief account as to who is SpayVac for wildlife. So the story begins in the early 90s when the Department of Fisheries and Oceans in Canada had a desire to reduce the numbers of gray seals that gather by the thousands on Sable Island, Nova Scotia every year. And there was a group of scientists at Dalhousie University then that were tasked with tackling this problem. And the key criteria were that the vaccine had to be long-lasting with a single dose. So the Dal scientists explored the use of liposomes as a means of enhancing the immune response. And for those of you who don't know about liposomes, I'll go into that in a little bit. And the result then was SpayVac, which is proven to have long-lasting single-dose characteristics in several species. And the company that I now am affiliated with, SpayVac for Wildlife, was formed last year with a couple of other biologists. So the Sable Island experimental design was really quite robust. There are a number of animals there had been a number of studies of these seals before, and so a number of the animals were already freeze branded and of known age. There were two, well, first of all, the animals that were selected were nursing, which meant then obviously they were proven breeders. There were two treatment groups, one receiving SpayVac and one receiving placebo, which is just the liposomes and the adjuvant. And the SpayVac treatment was just 100 micrograms of PZP, the liposomes, and Freund's complete adjuvant. And in the first year, year zero, of course, all of them were breeding. That was the criterion for selecting them. These animals come out onto the beaches at Sable Island, and they're there for only a very brief time. During that brief time, they give birth and nurse their pups and mate. And that all happens in about 2 weeks. So there was no expectation that in that first year that there'd be any effect because the mating would have occurred so quickly after the inoculation, or either before or after the inoculation. There simply wouldn't have been time for antibodies to develop to a point where they'd have a contraceptive effect. And yet, there was some degree of an effect. And my suspicion is that the zona pellucida surrounding the ovum, which develops in seals to the blastocyst stage, at which point development is arrested and implantation in the uterine wall is delayed. And there may have been something that happened during that period of delayed implantation that actually affected whether or not the blastocyst ultimately implanted. But then going on out to year 2, 3, 4, 5, 6, or 5, and then 10, you can see that something on the order of 70% of the females returned, of the treated females returned each year, but only a very small percentage of the treated females returned with pups. So it was a consistent high degree of efficacy. So all this happened before I got involved in the project. The work was published in 1997, and I came along then in 1998. In fact, almost exactly 20 years ago today, and was told that there was an island nearby where there was an overabundance of deer. We estimated on this 800-acre island, I think it was, that we had about 1,400 deer. And I think when I ultimately counted the number of deer in this particular spot, it was almost 100. And that's just a tiny part of the island. So very clearly, there were a lot of deer. So, going back 20 years, my thought process was, what can you do to contracept these things? At that time, I considered the potential of using hormone implants and quickly rejected that because of potential effects on the food chain and on the subject animals, and then the relatively short duration of efficacy. Surgical sterilization, I reckoned, would be too invasive and too expensive. And then about that time, I learned about immunocontraception using PZP vaccines. And here, of course, there were no food chain effects and minimal effects on the treated animals. So with the PZP vaccines, the target tissues are relatively specific, and so for the most part, non-target tissues are unaffected, certainly not toxic. Side effects appear to be minimal. With females, there are repeated estrous cycles, and it does extend the breeding season. I've seen male deer mount contracepted females into April. And it's primarily the younger males. The dominant males basically are active in the rut, and then they retire. And you'll see the younger males attending females subsequently. And at the time we were beginning this work, I guess by the time we were beginning this work, the vaccines had been in use for, I guess, really just about 20 years, and were generally regarded as being safe and humane. So as we already know, the zona pellucida is the outer layer of the mammalian ovum. Sites on the zona pellucida are critical to sperm binding. The zona pellucida is shed before implantation, and it's very similar amongst many mammal species, which is why you can take pig zona pellucida and inject it into a mare or into a doe, and the resulting antibodies will in fact bind to the surface of the treated animal's ovum. So this is a rather messy table, but I'll try to go through it sort of systematically. The very first trial, can you hear me all right without a microphone? The very first trial on James Island, British Columbia, went for just 3 years. And in that 3 years, we had no pregnancies whatsoever from a single dose of vaccine. The untreated controls were about 96 to 97% pregnant. Amazing. We went then to a trial on white-tailed deer. Oh, and I should say used 100 micrograms of PZP. We used Freund's complete adjuvant, and Vaximax is one of the forms of liposome that we used, and I'll explain a little bit more about that in a few minutes. Then we had a call from the Johnson Space Center, and they wanted to see if we could do something with the deer at the Johnson Space Center. So again, we used this time 200 micrograms of PZP, Adjuvac as the adjuvant, and Adjuvac is a dilution of an existing vaccine against Johne's disease in cattle. And Vaximax was the liposome type. So in the first 2 years, we had no pregnancies in the treated animals, and in the third year, 1 out of 8 animals was pregnant. Well, we had between 78 and 100% pregnant in the control animals. At Penn State University was a trial of captive animals, and there were 5 just 5, it was a small groups, small pen groups. The trial actually went out to 5 years, and there were no pregnancies over those 5 years. In another trial, but with a different form of the liposomes, we had only 4 deer. We had good performance out to year 2, but in year 3, 2 of the 4 animals became pregnant. White-tailed deer, Fripp Island, South Carolina, that Alan was referring to. Again, we used 200 micrograms of PZP, Adjuvac, and the Maxivax liposomes. Perfect performance in year 1, but in year 2, a quarter of the animals was pregnant. And we don't have data beyond just year 2 for that study. And with white-tailed deer also on Fripp Island, using Depovax, again, the performance wasn't as good as we had hoped for, but there was still some effect. One of the things that I think complicates the Fripp Island story is that the animals there were undernourished, and I think their ability to mount a strong immune response was probably hampered by their nutritional status. Now, we did another study with black-tailed deer in British Columbia, and this study went out for 6 years. We had a total of 12 deer. Not all of them were treated in the first year, and we had no pregnancies until we got out to year 6. For control, the owner of the property knew the animals as individuals, and in the pre-treatment period, there were 24 doe years, and in those 24 doe years, those does produced 34 fawns, or 1.4 fawns per year. After treatment, we had 54 doe years, 1 fawn, and you can see the differences. You don't need to do any statistics on that to know that it's different. So the key to all this are the liposomes, and if you can imagine, I always try to describe them as, imagining a small microscopic onion, and with the antigens and the adjuvants sandwiched in between the layers of the onion. So they're very small, multi-layered vesicles. They're comprised of cholesterol and lecithin, and they result in a prolonged release of the antigens. The result is a rapid response in antigen buildup, and they're long-lasting after just a single dose. In all the studies I'm talking about here, we've never boosted. So just a comparison with ZonaStat and SpayVac, both use PZP antigens, both use an EPA approvable adjuvant. We're tending to stick to Adjuvac. ZonaStat, I think, is using modified Freund's. SpayVac includes using a liposome technology which is missing from ZonaStat. we get a long-lasting result with a single dose, and ZonaStat requires boosting. One of the consequences is that we can do this with a minimal disturbance to the animals. We only have to treat them once, and they're contracepted for an extended period. And because of that, it's more economical to implement. You don't have, it costs money to have people out catching and darting animals. Now, I mentioned there are two different forms of the liposomes. Vaximax is the original formulation. It's the one with which we've had most experience. We've normally received it in preloaded frozen syringes. It needs to be kept frozen until it's used. And the emulsion in the syringes is roughly the consistency of mayonnaise. Mayonnaise, of course, is a familiar emulsion that we all know about. Depovax is a more advanced formulation, but we have much less experience with it. It's provided in 2 vials. One vial has sort of a cake of the antigen and liposomes, and the other vial is mineral oil and adjuvant. And you simply take the contents of the mineral oil and mix it with the cake of antigen and antibody and liposomes, and it's much easier to handle. It has to be kept cool, but not frozen, and it's much more amenable to dart delivery because it's the consistency of light mineral oil. So we see some advantages to SpayVac, generally less disturbance and less risk to animals and to people who are doing the work. There's a reduced potential for late breeding and late fawns because it lasts longer. Improved feasibility, you don't have the same need to try to relocate previously treated does to administer booster doses. More economical because of reduced labor costs. And the company that was spun off from Dalhousie University has its focus on cancer and infectious disease vaccines. So they're continually working to improve the basic technology. And we have the ability to piggyback on their advances. And similarly, they also have contract manufacturing organizations that physically produce the vaccine, as is typically done in the pharmaceutical industry. And we have the ability, if we get to the point where we need to make large quantities, of being able to scale up quite easily. So late last year, Ursula Beckert and a fellow named Tom D'Orazio and I formed SpayVac for Wildlife to further advance contraceptive vaccines for application to control overabundant feral and native animals. Tom, who's the president and CEO, has more than 25 years experience with major pharmaceutical companies and with startups. And I've known for a long time just how limited my knowledge of business was, and Tom has definitely proven me correct on that point. He knows how to do things that I would never have thought of. Ursula is a veterinarian with a PhD in reproductive endocrinology, and she and I have worked together for 15 years or so on horses and elephants. And then I'm the vice president of operations. So, questions, please.